Today's lecture will cover Chapter 15, and we're going to talk about gene mutation. Chapter 15 also talks about DNA repair, but we're going to focus on the mutation today. So in today's topics, I'll cover gene mutation, how we classify different types of mutations. I'll talk about the difference between spontaneous mutations and induced mutations. And finally, we'll go over what exactly the Ames test is and how we utilize Ames testing to identify mutation. Right? So mutation is an alteration in the DNA sequence, and that alteration can be a single base pair, say substitution, in which we take an adenine and maybe substitute it with thymine. It could be a deletion or insertion, where we're either taking out a base pair or putting in a base pair, or it could even include major alterations in the chromosomal structure, similar to what we talked about in our previous chapter on chromosomal aberrations. Furthermore, mutations, they can occur in somatic cells. So a somatic cell is basically any of the cells in your body that is not a sex cell. Okay, so not an egg or a sperm. All of those cells would be somatic cells. Obviously, these cells are not passed on during sexual reproduction, or they could occur in a germ cell. And if a mutation occurs in a germ cell, these are our gametic cells, like an egg or a sperm, then these would be passed on to future generations. Mutations are also classified as what we call spontaneous, meaning that they happen naturally and they're random. They are usually linked to normal biological or chemical processes in the organism that occur during replication. And I will present some data in a couple slides that kind of proves this notion that our mutations are indeed spontaneous. A couple other notes that we should make about mutations is one, that they are actually exceedingly rare in most organisms. So we have a very low rate of mutations. Every time we copy, there's a small number of base pairs that may acquire a mutation, but it's very small. Those rates, they can differ between different organisms. And even within the same organism, rates can vary from gene to gene. So if we look at spontaneous mutation rates, this is in different loci in different organisms. So here we're looking at, like say in a bacteriophage, the character state or the trait would be lysis inhibition. So going from a mutant back to a wild type, we find that mutation occurs in roughly 1 × 10⁻⁸ times. So a really small percentage of organisms have that. We can see though, hey, at a different gene within bacteriophage, they have an even smaller mutation rate. So mutation rates are not the same for each gene in an organism. Okay, it largely has to do with because most mutations occur during replication, the sequence of that region of the genome that's being replicated. Furthermore, if we compare mutation rates, say in Homo sapiens, so here's Huntington's disease, hemophilia, here's the mutation rates. Those mutation rates are not the same. They are a little bit larger. They're not quite as small as, say, the mutation rate in the bacteriophage. So again, we have differences between organisms. Okay, so our two types of mutations that we can have. First one is that's spontaneous. Okay, a mutation that happens completely naturally, it's random. The process that results in the mutation is either a biological or a chemical natural process. The other type of mutation we can have is what we call induced mutations. And these ones result from the influence of an extraneous factor. It could be natural or it could be artificial, but it's extraneous. So things like radiation, UV light, synthetic chemicals, carcinogens, mutagens, those would be induced because they aren't just occurring randomly in the natural environment, rather they're occurring because of an extraneous factor that is increasing the likelihood of that mutation. Now this brings up a really unique question, namely, Are mutations, do they arise randomly or are they directed by the environment? And when I say directed by the environment, what we really mean is there a selective pressure in the environment that causes the mutation to occur? Now, this is different than just being induced, saying, hey, you have like a mutagen that causes mutations to occur at a higher frequency. What we're saying is there a selective pressure that causes a very specific mutation to occur? To test this, there was a number of experiments that were done, but the one I want to talk about is the Luria-Delbruck fluctuation test. So these were two researchers that wanted to answer this question. Are mutations adaptive, meaning are they directed by the environment, or are they random? And so what they did was they grew up a bunch of E. coli in a large flask, okay, to where they got lots and lots of E. coli. Okay, sorry, my flask looks pretty janky. I'm drawing with the mouse. Okay, so they had a uniform concentration. They also grew up E. coli in, so they also grew them in test tubes. I'm just going to draw 4. Same thing, and they grew them up to where they had the same concentration of E. coli growing in this. Now these E. coli were just growing in a medium with just themselves. And as they're growing, what we mean by that is that they're dividing and you're having lots of generations. So with each generation, there's a chance that a mutation could be acquired and be passed on to subsequent generations. Then what they did, from the flask culture, they plated set amount of those E. coli onto a medium like agar that contained T1 bacteriophage. So this is a virus that infects E. coli and kills it. And so essentially you're creating a medium that the only way E. coli can grow is if it has acquired a resistance factor. And the initial E. coli that they put in here were not resistant to T1 bacteriophage. And they incubated these and allowed them to grow, and they would get a few colonies that would grow, meaning those few colonies were the ones that had acquired a mutation with a resistance factor. Now, with the test tube ones, they did the same thing, but each test tube got its own petri dish. And again, they all had T1 bacteria or bacteriophage on there. And again, they would get some growth from those that were resistant. This is what the results look like, right? So this group right here is their sample culture. That's the control group. So I'm going to put a C for control. And this group over here are the different cultures. And so what we see is for the control group, there was an average number of colonies, about 16.7, and the variance, it wasn't, it was 15 colonies, but that's not that high of a variance, okay? So pretty uniform, which is what we would expect because they're all the same bacteria that got put on there. That's why we used it as a control group. They all came from the same culture. So if there were a few bacteria in here that required resistance and they're being mixed and they start dividing, and you would have relatively the same proportion of resistance bacteria spread on each plate, so you'd expect to have about the same number of colonies on each plate. Now there's some randomness to the sampling that explains the variance, but for the most part it's the same. But on the ones that were different cultures, they found an average, a mean number of 26.2, but the variance was much, much higher. So 2,178 was the variance. So you can see, like, some of them only had a few, some of them had a lot, like 165 on this. And so from this data, what can we conclude? Are mutations random, or are they being directed? Well, if they were being directed, what that would mean is that as soon as the E. coli is put on this medium with the T1 bacteriophage, that the T1 bacteriophage infecting those bacteria would cause them to then acquire the mutation. And if you have the same amount put on the same thing, we would expect these different cultures to be very similar to the control. It's basically saying at this point, you need to mutate. You would takes a little bit of time to mutate, and you'd have the same on those. So the fact that we have all this distance variance means for each one of these, it's just a random chance that one of the bacteria in these different cultures acquired the mutation. They're not acquiring it at the same time, they're acquiring it at different times. So this really kind of gives a lot of evidence that mutations are random. They are not adaptive, they are random. Okay, so let's talk about how we can classify mutations. We already talked about spontaneous first induced. We can also have what we call somatic mutations. Those are the ones that occur in the germ cells, or germline mutations that occur in the gametes. We can have autosomal mutations. So if a mutation happens on the chromosome or a gene that's located on their autosomes, that would be an autosomal mutation. Or we can have X-linked or Y-linked mutations if those genes are associated with the X and Y chromosome. Our types of mutations can be classified as either a point mutation, sometimes we call these base substitutions, in which we change one base pair, so one nucleotide, to another. And as we talked about in the previous chapter, on transcription in the genetic code, that can sometimes result in a new triplet code for an amino acid. If that occurs, if that one base pair being changed causes a different amino acid to be coded, then we call that a missense mutation. Okay, if that change in base pair results in a stop codon being formed, we call that a nonsense mutation. Right? And obviously we're going to have translation or transcription is going to get terminated. Not transcription. Translation will get terminated. We'll still transcribe that codon. But when we go to translate, it's going to get terminated prematurely. We won't get the full polypeptide. And finally, if the triplet code still codes for the same amino acid, so we had a base pair substitution in a coding region, but it doesn't change the amino acid. Let's say it happens in that third position that sometimes is variable because of the wobble hypothesis. We call that a silent mutation because it doesn't result in a change in amino acid sequence. Furthermore, base substitutions can be classified as either a transition or a transversion. So a transition is where we replace a pyrimidine with another pyrimidine. So that would be like going from cytosine to thymine. Okay? Remember, these are normally what these guys pair with. I'm going to draw the hydrogen bonds in. Okay, or if we had to switch from adenine to guanine, that would be a purine to purine or a pyrimidine to pyrimidine. Those are transitions. Okay, transversions are when we have a purine and a pyrimidine get interchanged. So that would be like a C to an A, G to a T, or even C to G, A to T. Those would all be transversions. Just as review, because we talked about this with genetic code, a frame shift mutation results when we have either an insertion or a deletion of a base pair. And this can be severe because it can cause our entire frame shift, or our entire reading frame to shift. So again, if we think about the genetic code, it's read in triplet, so it's like 3-letter words. If our code was translated and it basically was the cat saw the dog, right? These are all of our base pairs, THE, CAT, and These are all codons. They make sense. Well, if we did a substitution, let's say we had a point mutation, we could have something that says like the bat saw the dog, or the cat saw the hog, or the cat sat the dog. Okay, we've changed things. Maybe these are changes in amino acid, but it's still, we haven't changed our reading frame. But let's say we have a deletion. We lose the C. Well, now suddenly we don't have something that makes sense, right? We have the at, ought, head, og. Or if we insert one, the semmatisia with the ego D. You can see how frame shift mutations are going to change everything afterwards. Furthermore, we can classify mutations by what they do, right? We could have a loss of function mutation. What's the phenotype of that? Well, we lose the function of that gene. We could have a gain of function. We could have something that previously did one thing and now it does a completely different thing. We can have visible or what we call morphological mutations. We could have nutritional. These tend to be biochemical mutations, so that it has to do with biochemical pathways. Behavioral mutations, regulatory mutations would be not in coding regions, but a mutation in a regulatory region for a gene, so gene expression gets changed. We could also have what we call neutral mutations. And this will make more sense when we talk about population genetics, but in a neutral mutation, we don't increase or decrease the overall genetic fitness of that individual. And I'm just introducing this now because when we get to population genetic, we'll talk a lot about neutral mutations. Right? They don't affect, gene products or gene expression. So in some ways, silent mutations are neutral mutations. Okay? The majority of mutations that occur in the genome are neutral mutations because a lot of mutations aren't tolerated very well. If we change the amino acid and the protein doesn't function, that could lead to a lethal trait. Now, how do our mutations arise? Well, they arise from replication errors and from something called base modifications. So during DNA replication, occasionally DNA polymerase is going to insert the incorrect nucleotide. And when it does this, if it doesn't detect it and go back and correct it with other DNA polymerases, like DNA polymerase 3, then these types of errors are going to persist, and then we're going to have a mispairing, and that leads to a point mutation being occurred during the next round of mutation of synthesis. Additionally, we can have where a DNA strand where it creates a loop, right? It loops out, becomes displaced during replication, and if that happens, the DNA polymerase can quote unquote slip. It kind of falls off and it reattaches itself after the loop. And that can cause things like insertions or deletions. We saw that when we talked about the chromosomal mutations. Okay, and a lot of time where we have replication slippage is in regions of DNA where we have these repeat sequences over and over again. We see that in Fragile X disease, it's on the X chromosome, and Huntington's disease, same thing. Okay, so that's DNA replication errors. We can also have what we call base modifications. So in purines and pyrimidines, if we start thinking about their chemical structure, you may remember from general chemistry, from inorganic chemistry, that when we have these types of hydrogen and covalent bonds, so when we have this covalent bonding, sometimes covalent bonding will shift, electrons will shift their valences as they try to enter into a stable valence, and so we can have different isomers or what we call tautomeric forms. So for instance, thymine is generally found in the keto form. However, we can shift some of these electrons around in which we have a covalent bond and a double bond between the carbon and the nitrogen and a single on this oxygen, and that puts it into what we call the enel form. Now, this is usually not that stable. It likes to be in keto, but it's times where it can shift into that, depending on the stress that it's under. And in the enol form, you'll see that it can pair with guanine, whereas normally it pairs with cytosine. Same thing can happen with cytosol. or cytosine, sorry. Right, cytosine can go into its amino form, in which, again, we're shifting. Instead of having the double bond here, we double bind to our nitrogen here on this amine group, and it'll bind with adenine. So these are tautomeric shifts. It's a single proton shift in the molecule, and it puts it into a different form, and it can allow for mispairing to occur. So what does that look like when we have a tautomeric shift? Okay, so let's say we're replicating our DNA. We break our double strand. This is generally when the tautomers occur. When we go to single-stranded, in this particular adenine, we have a tautomeric shift. So it shifts into its amino form. And then when we start doing our semi-conservative replication and making our strands, so this strand doesn't have any mutation, but here, this tautomer, instead of binding with thymine in the amino form, it binds with cytosine. And so now we have this mispairing. So in the next round of replication, what happens? Well, we go single-stranded, and because generally these base pairs don't like to be in these tautomers, and the only time it happens is when they're not being bound up, it tends to shift back. So that adenine shifts back into its regular form. And so when we go into the next round of replication and we make the complementary strand, now we do the base pairing, and you'll see we pair again with thymine, but in this cell, that cytosine remains, and it's going to normally pair with guanine. And so now we have a cell in which we have cytosine guanine over here, and over here we'd have adenine thymine. Some of the most common causes of spontaneous mutations are from DNA base damage, where we damage the base itself. First of that is called depurinization. And in depurination, we lose one of the nitrogenous base. It's usually a purine that's lost. And instead, we just have the intact double helical model, but we have no base pair. So we have this hydrolysis reaction. This glycosidic bond breaks, and we end up with what we call an apurinic site. And if we don't repair it, then we can, during replication, polymerase could stick any random nucleotide in that spot. Another one would be deamination. In deamination, the amino group, usually in cytosine, can also happen in adenine. It gets converted into uracil. And so when we get a cytosine that goes to uracil, we get an adenine, it's converted to hypoxanthine, and then we get these mispairings. So instead of having an adenine to thymine, which is what we normally have, we could get it converted to where we now have a guanine cytosine in that original place. So we can see that right here. So here's our cytosine. We lose this amine, hence the name deamination. It results in now a uracil, and that uracil binds with adenine. And so we get that AT. We could also have an adenine that loses its amine, and it becomes this hypoxanthine that can bind with cytosine. So we get this mispairage. And then this would then be replaced in replication in the next time with a guanine, right? Cytosine would pair with guanine, adenine would pair with thymine. Another form of spontaneous mutation is through oxidative damage. So you may have heard people talk about how the fact that it's not good to have diets that are in low, that don't have a lot of free radicals, because free radicals, right, those can cause oxidative damage. It's a normal cellular process. It's where we have high energy radiation. When we start releasing radical groups out, like our superoxides or our hydroxy radicals, these can sometimes break or cause our bases to change their chemical composition. And we can lose bases. We can also have single strand break, where we break some of those glycosidic bonds, and those can cause mutations. Now, on the other hand, if we talk about induced mutations, These arise from DNA damage caused by chemicals or radiation, so by foreign substances. We call those substances mutagens. Okay, they can be natural or artificial agents, but they cause mutations to be more likely to happen than just through tautomeric shifts or deaminations or depurinations. Some examples of this would be things like fungal toxins, cosmic rays, ultraviolet light, industrial pollutants, medical x-rays, even the chemicals in tobacco smoke. What generally happens when we have these mutagenic chemicals is that they cause base analogs. So these are similar to tautomers, but they're being forced, or they act similar to a tautomer, let me say it that way. But it's because we have these new things in. So for instance, 5-bromouracil is a base analog of thymine. So if we bring in this bromine, it can cause thymine to turn into this uracil, and in this form, it can bind with adenine, just like thymine would, in its keto form. But in the eno form, it's going to bind with guanine, and it increases the frequency of a tautomeric shift. So tautomeric shifts can happen normally, but again, they really don't like to happen that often, so it's pretty rare. But base analogs go into tautomeric shifts at a higher frequency. Hence, we say that chemical is mutagenic. Okay, in this case, it also 5-bromouracil is really sensitive to UV light, so the UV light causes it to do that tautomeric shift. We can have things that what we call alkylating agents, in which they donate an alkali group, so to their amine or keto. So here's a guanine. We have an alkylating agent. This is EMS, and you can see it adds on this alkali group. And now we have a 6-ethylguanine. Guanine would normally pair with cytosine, but now it'll pair with thymine. We can have things called intercalating agents. These are chemicals that have dimensional shapes that squeeze themselves in between the DNA bases and break those hydrogen bonds. And so they can cause distortions in the DNA. And then this causes that DNA to unwind, and during replication, there's a lot more mutations that occur because the things are being pulled further apart. Some common intercalating agents are things like ethidium bromide. We use that to actually visualize DNA in gels when you do gel electrophoresis. A lot of chemotherapeutic agents are intercalating agents. You have things like adduct-forming agents or mutation-causing chemicals. Those will bind to DNA and they alter its conformation. So all of these induced mutations are chemicals or natural processes that change the chemical structure to such where we have a lot more tautomeric or a lot more base analogs occurring. So example, this would be acetaldehyde. We find that in cigarette smoke. HCAs, those are the cancer-causing chemicals we find in when you cook meat, they get released. All these things alter DNA's conformation. Things like UV radiation. Okay, UV radiation, we have high amounts of UV light. What that can cause is it can take, if we have two identical pyrimidines right next to each other, like in this case thymine, they can distort the DNA conformation such to where we actually start creating covalent bonds between those adjacent thymines. And that creates a little dimer and a kink in the DNA helix that can cause base mispairing downstream. Ionizing radiation, like things we get from X-rays. We know that X-rays, gamma rays, comic rays, that they're mutagenic. How do we know that? Well, people have done tests in which they've exposed, in this case, these were probably rats being exposed to radiation, X-rays, and they found that, yeah, you get a lot more X-linked recessive lethal gene mutations occurring, the more radiation, the more X-ray doses. That's why when you go to the doctor, you get an X-ray. They tend to not like to do lots of X-rays really close together because it can cause mutations. Last thing I want to talk about is the Ames test. So the Ames test is the test we use to assess how mutagenic is a compound. And we use this with Salmonella typhimurium strains, and they're selected for their ability to be able to reveal the presence of specific types of mutations. So we use this with any chemical. If you're going to create a product and you want to say, hey, it's non-carcinogenic, it's non-mutagenic, you need to do an Ames test on it. And the way you do that Ames test is, again, you take your Salmonella typharium and you grow a very specific strain of it that is not able, it's the His minus oxytroph, so it's not able to produce histine. Therefore, it needs to, in order for it to grow, you have to provide liver enzymes so that it can produce that histine. It needs that to grow. So you grow it up in a culture where it has that. Okay, you can also then grow it in the culture. You take that mutagenic compound. Okay, it also has those liver enzymes on there, and you put that mutagenic compound on a small filter disc, and you stick that on a medium. Okay, that contains growth medium, but it doesn't have the liver cells. Okay, so what that means is when you put these on here, when you then plate the oxytrophs, you put the oxytrophs here, you put the oxytrophs on there, you spread it. We are only going to grow typharium on this agar if there's a back mutation that allows it to become spontaneously His positive, meaning it can produce its own histidine, so therefore it can grow. And that we know that's going to occur. There's a few growing right now in here that are likely to generate that, okay? They're going to have back mutations. When we put it on here with this plate, if we see a higher number of colonies around the mutagenic filter than on the plate that doesn't have it, that tells us that, hey, look, more mutations are occurring when we grow the bacteria in the presence of that compound. That probably means, and again, you know, the His autotroph would be in here too. That probably means that it's a mutagenic compound. I mean, not probably, it does. We have evidence. More mutation is occurring. The mutation rate is higher in the presence of the mutagenic compound than not.